Welcome everyone. Today we are going to learn about sodium calcium antiporter. But before I begin, we have to understand 4 important points. The first point we have to understand, that is downhill movement. Downhill movement. Second important point is uphill movement. Third point is chemical concentration gradient. Chemical concentration gradient. And the last point we have to understand, that is membrane or electric potential. Or we can say the voltage across the plasma membrane. So at first we have to understand these 4 important points, then we will understand the mechanism of sodium calcium antiporter, how it works. Okay, so at first I will discuss about what is downhill movement and uphill movement. Suppose this is an example of hill, and we know that if someone try to come down from a hill, it feels easy, it requires less energy and less effort, so we call it downhill movement. And the opposite will be true, that when someone try to climb up the hill, that requires more effort, more energy, so it is uphill movement. The same thing is true for molecules also. In case of downhill movement, molecules actually move from higher to lower concentration, and during this transport it does not require any energy output, because this process is thermodynamically favorable. So that's why this process is called passive transport. Passive transport. Okay. So this all 4 factors are background to understand the mechanism of antiporter. Now, during uphill movement, molecules move from lower to higher concentration, and that's why it requires energy input from the outside, just like during the uphill movement of if someone tries to climb up, then it requires energy. So it also requires energy output or input. That's why this process is called active transport. This process is called active transport. Now, in my previous video, I have mentioned that under the passive transport, that means during the movement of molecules from higher to lower concentration, there are 3 occasions when passive transport we see, that is simple diffusion. During simple diffusion, molecules move from higher to lower concentration. It also occurs during uniporter mediated transport, and during transport through the channels. So there are 3 occasions when molecules move from higher to lower concentration, but here we have to understand that during the simple diffusions, no transport protein is required. That's why we exclude this mechanism from passive transport. But in simple diffusions also, molecules move from higher to lower concentration, but here that does not require any transporter proteins. The molecules actually directly transport or actually cross the plasma membrane directly without the help of any transporter proteins, these simple diffusions. But in case of uniporter and channels, these are the proteins, transporter proteins, that helps. In my previous video, I have already discussed about one example of uniporters called blood proteins. These proteins helps in the import of glucose from blood. And channels, we know that through the channels, ions and small hydrophilic molecules, water dissolved molecules can pass through the channels. So here in 3 examples, molecules move from higher to lower concentration. Now let's take an example of active transport. Active transport. Here also during 3 occasions active transport occur. First is, we call it coupled or co-transport. Now, why it is called coupled? Because here two molecule or one molecule and one ions move simultaneously. They are obligated to move simultaneously because no one can move without the other. So both molecules or ions actually move with the during the movement or transport. Now this coupled or co-transport could be divided into two categories. One is called Antiporter mediated coupled transport, Antiporter, and another is Antiporter. In my previous video, Antiporter, I have discussed about sodium and glucose Antiporter. I have already made separate video on sodium glucose Antiporter, but today we are going to learn about this Antiporter mediated transport, specifically sodium and calcium Antiporter. Now, in other two cases like ATP driven pump and light driven pump, in both these cases active transport occurs. Here ATP provides the energy and here solar energy provides the energy, and all these three cases molecules move from lower to higher concentration. So we have to understand that it is not necessary for the molecules to move only from higher to lower concentration. Cells requires, it's the purpose of the cells, it requires sometimes that molecule should move from lower to higher concentration, so that it is called active transport, and it actually requires, and for that they have different energy source. Now in case of Antiporter, I have mentioned in case of sodium glucose how this active transport occurs. We will also explain how Antiporter works, because here also one molecule will move uphill movement, show uphill movement, and another molecule will show downhill movement. And in this case, in coupled transport, this downhill movement of one molecule actually provides the energy or release the energy for the transport of other uphill movement, that molecules that showing uphill movement. So I will explain here this mechanism. Now we have actually covered, understand this downhill and uphill movement. Let's now talk about the chemical concentration gradient. So I have to remove this. Now let me explain what is the role of chemical concentration gradient and membrane potential. To understand this, I have to draw the lipid bilayer of plasma membrane. So let me draw the lipid bilayer. Suppose this is the lipid bilayer of the plasma membrane. So it indicates the outside or exterior of the cell, and this is the cytosol. So we are going to understand two factors. I have mentioned the third important factor is chemical concentration gradient, and the fourth point is membrane or electric potential. In short, we can say the voltage that present across the membrane, okay, across the lipid bilayer. So these 3 and 4, the last two factors, are actually called the driving force of the transport of the molecule. So this acts as a driving force. Okay, now here is a discrimination between two types of molecules. Those molecules carry no net charge, that means those molecules are uncharged. Remember this, those molecules are uncharged. This chemical concentration gradient, I am just writing here CCG, chemical concentration gradient. This is the only driving force for uncharged molecules. But in case of charged molecules and ions, in case of this category of molecules, both the chemical concentration gradient and membrane potential or electric potential actually togetherly work, and we call togetherly these two factors, togetherly we call it electrochemical gradient. Electrochemical gradient. Okay. So for uncharged molecules, only chemical concentration gradient is important, is the driving force. But for charged molecules and ions, both the factor works togetherly, and we call electrochemical gradient. In case of ions, specifically ions, there is a terminology we call the same, these two factors that cause the driving force, we call it ion motive force. ion motive force. So whether you can use this terminology electrochemical gradient or ion motive force specifically for ions. Okay, so let me explain one by one how chemical concentration gradient works and how membrane potential works. So to understand chemical concentration gradient, we have to take an example of uncharged molecules. One example of uncharged molecule is glucose. Okay, glucose don't carry any charge. So let me draw one uniporter through which glucose actually transport inside the cell, that is called glut proteins. GLUT. Through this glut protein, glucose molecules actually enter inside the cell, so import occur. So glucose present outside. Suppose this outside is the blood. So if this is the blood, then this glucose actually entered inside the cell. Okay. Now one thing we have to understand that is the concentration of glucose outside or the blood is high. Most cases, in most situations or most cells, the concentration of glucose is high in the blood or extracellular fluid than the inside. So glucose concentration is lower inside. So that's why molecules like glucose, when move from higher to lower concentration, that is the downhill movement, and that occurs through the uniporter. Okay, we can draw the chemical concentration gradient by example by triangle. We can draw the chemical concentration gradient by a triangle. The base of the triangle, this is the base, indicates that here the concentration of the molecule is high. The tip indicates that here the concentration is low. For glucose, as because the glucose concentration is high outside, so we can draw the base outside and the tip of the triangle inside. That shows that glucose is higher in the outside. So by this way we can represent chemical concentration gradient. And here in case of glucose, this is the driving force of this molecule to get inside into the cells, and this is the passive transport. Now let me explain what is membrane or electric potential and how it works. Basically togetherly these two factors works. So how this membrane or electric potential generates? So there is two transport proteins present responsible for the generation of membrane or electric potential. One is called one pump, that is called sodium potassium pump. Sodium potassium pump. So this pump is actually fall under the ATP driven pump. So one ATP is breakdown that gives the energy to transport sodium and potassium. Now here we have to understand sodium ions, 3 sodium ions actually transported out of the cell, and 2 potassium ions are import inside. And as you can see, 3 positive ions are moving outside and 2 inside. That means in the exterior more positive ions will be present. So that's why it generates membrane or electric potential. So more positive ions will be outside than inside. So it will create a negative charge inside and positive charge outside. One thing we have to also understand that the sodium ions concentration will be remain high outside and will remain low inside, because this pump actually exporting sodium out of the cell. And the opposite will be true for potassium. The concentration of potassium ions will be low outside and will be high inside. And by this way, this sodium and potassium pump actually generates the ion concentration gradient for the sodium and potassium in the cell. So this pump not only generate membrane potential but also chemical concentration gradient. Okay, now this pump is actually ATP driven pump. Why? Because sodium, this pump works against the concentration gradient, because as I mentioned, sodium is higher outside, but this pump actually moving sodium from lower to higher concentration. That's why it is uphill movement. The same is true for potassium ions also. In case of potassium ions, this potassium ions is actually moving from lower to higher concentration. That's why it is a active transport, ATP driven pump. Now the membrane and electric potential are not generated by solely on sodium potassium pump, but there is another transport protein called potassium channel. these potassium channels are actually called potassium leaky channels. Potassium leaky channels, they are non-gated in nature, and through this, as because there is a high concentration of potassium inside, so through the channel potassiums actually move outside of the cells. As I have mentioned, that channels also show passive transport. That's why I have mentioned that these 4 factors are very important to understand this antiporter also. Now see, as because more potassium ions are moving through these channels, so more ions, positive ions will be generated outside. More positive ions will be generated outside than more negative. As because there is more positive charge here, so there will be lack of positive charges, so there will be more negative charge inside. That is the situations. Now, so this is all over the plasma membrane. I'm just drawing here on it. See, these two transporters make one potential. We call it resting membrane potential. Resting membrane potential, and this is not only for electrically excitable cells like nerves, muscles. Not only for that, all animal cells have the resting membrane potential, and that the value is generally minus 70 millivolt, but the value can vary from minus 20 millivolt to minus 200 millivolt, and that depends on the cells and the organisms. Generally, the value is minus 70 millivolt. This is the resting membrane potential. It is true for all animal cells because these two transporters works in every animal cells, and it establishes this concentration gradient. See here, I have mentioned that sodium potassium concentration gradients are already generated by this pump, and also this membrane or electric potentials are generated by this sodium potassium pump and potassium leaky channels. So here the membrane potential, or we can say these electrochemical gradients are generated. Now how this electrochemical gradient actually works, or as a driving force, let me show you. Suppose the example of sodium and calcium antiporter. Take the example of sodium and calcium antiporter. So this is the sodium calcium antiporter. I'm just showing here the movement of sodium to explain it, how it occurs. See, I have mentioned that outside sodium is high, so I will draw the triangle. The base will be outside and the point will be inside. Okay, because the base indicates the higher concentration. So outside sodium concentration is high and inside sodium concentration is low. Now, not only this electrochemical concentration gradients helps to import, so sodium will move inside the cells because outside sodium concentration is high, inside is low, so it will move from higher to lower concentrations, that is downhill movement. But here not only this chemical concentration gradient is actually helping the movement, but also the membrane potentials is working under beneath. How? See here that positive charged ions will be more attracted toward the negative charged area. So this will also help in the import of the sodium inside the cells. That's why it is called electrochemical gradient, or for ions particularly we call it ion motive force. Okay, so here ion motive force means concentration gradient and the membrane potential that is generated by sodium potassium pump and potassium leaky channels during resting membrane potentials. So chemical concentration gradient and both these membrane potential works together to import the sodium. Now here in this antiporter, sodium calcium antiporter, the movement of sodium is the downhill movement. Okay, here from the higher to lower concentration the sodium is moving. So now let me explain how the sodium calcium antiporter works. The outside concentration of sodium is about 145 millimolar and inside is 12 millimolar. So as you can see, there is a 12-fold change or 12-fold difference in the chemical concentration gradient. And as this membrane actually across the membrane there is a potential or voltage changes, so the movement of sodium is actually favored. But in this case the calcium actually moves from lower to higher concentration. As you can see here, in case of calcium, the inside concentration is 10 to the power minus 7 molar and outside is 10 to the power minus 3 molar. So the net difference is here approximate 10,000 fold. Remember this. So there is a huge difference because calcium is very important for our cells. It acts as a second messenger and also helps in the functioning of many physiologic functions like neurotransmitter secretions, muscle contractions, and in the cell signaling also. So these calciums actually are kept in low concentration, very low concentration inside the cell, inside the cytosol. So as you can see, there is a 10,000 fold change. Now see here, I have mentioned that during this antiporter and symporter, both these cases, these are the active transport. So in here, one molecule actually moves downhill movement, show downhill movement, and another molecule show uphill movement. In case of sodium, it shows the downhill movement because the chemical concentration gradient look like this. So if I draw the triangle, the base will be the outside and the tip will be inside because the sodium concentration is outside is more. So the sodium is actually moving from higher concentration to the lower concentration. That's why it is downhill movement. So I can write sodium shows the downhill movement. But opposite is true for calcium. The concentration of calcium is higher outside, okay, than inside. See, the opposite is true for calcium. That the concentration is higher outside, lower inside, but the calcium is moving from lower to higher concentration. As you can see, from inside to the outside. Now, why cells require this type of movement? Because as you can see, this is actually uphill movement. So calcium is showing the uphill movement. Okay, it is showing the uphill movement. And this downhill movement of sodium is actually release energy. I have made a separate video on sodium glucose symporter, and that video I have shown how this chemical concentration gradient and membrane potential together the electrochemical gradient generate energy. So this movement of calcium is actually important. Why it is important? Where this sodium calcium antiporter stands in our physiologic conditions, because it has a particular functions. The functions depends actually in the cardiac myocytes. Cardiac myocytes. Okay. Now what happens here? Scientists have found that if we inhibit this pump, by chemicals called cardiac glycosides. These chemicals are basically plant alkaloids. Examples like ovin and digitalis. These two are plant alkaloid that can inhibit the sodium potassium pump. And as I have mentioned, the sodium potassium pump actually generate the ion concentration gradient of sodium and potassium. So if we inhibit this pump, what will happen? This membrane potential and the sodium concentration gradient will not be there. Reduce. The sodium concentration gradient will be reduced and this whole membrane potential will be disrupted. And that will decrease the movement of sodium from outside to inside, and that will also decrease the movement of calcium from inside to the outside. And what will happen? The concentration of the calcium will start to increase inside. Okay, so if we inhibit this pump, the calcium concentration will start to increase. And what we know about calcium increase? We know that if in the muscle or this cardiac myocytes means it is also muscles. So in the muscles, whether it is cardiac muscles or skeletal muscles, increase in calcium concentration, what causes it? It causes contraction. Okay. And we know if the calcium concentrations decreases, what will happen? The muscle will relax. So relaxation occurs. So basically, this antiporter plays role in the relaxation because it decreases the calcium concentration in normal situations. But when we actually treat cardiac myocytes or cardiac muscles with this type of chemicals like obin and digitalis, these two chemicals, they actually inhibit this pump that disrupt the electrochemical gradient of sodium, and that actually hampers the functioning. It will perform less efficiently, so calcium concentration will increase, so it will contract. So these drugs are used in the treatment of heart failure. Now, this contraction of cardiac myocytes and also the muscles actually go through one process that's called, this is very important, basically action potential actually arrives at the cardiac myocytes and then contraction occurs. This is the second phenomena. The first is action potential arrives at the cardiac myocytes. So this process arising of action potential and causing of contraction we call excitation and contraction coupling. Remember this process is very important. Excitation contraction coupling, that means when action potential, that means excitation occurs, contraction will occur. This follows togetherly in case of cardiac myocytes also. Now, to understand how sodium calcium antiporter works, we have to understand this excitation contraction coupling. That means at first we have to understand the action potential of cardiac myocytes. Then we can understand how contraction occurs in the cardiac myocytes, and then we can relate the presence of sodium and calcium, or the importance of physiological importance of sodium calcium antiporter. Because just by knowing that sodium actually entering inside and calcium moving outside, it doesn't mean how it works and where it stands in the physiological conditions. So in my next video, I will talk about more details on the excitation contraction coupling of cardiac myocytes, the action potential of cardiac myocytes, and how contraction occurs. Because we have to understand that the action potential of cardiac myocytes is somewhat different from nerve action potentials. So I will explain in the next video. Hope this video will help you. Thank you for watching and always keep learning.